Welcome back to Day 2 of Advances in Drug Discovery and Development 2024. We're incredibly excited to have you all here, and we can't wait to see the incredible insights and connections that will emerge today. I'm George Hammond, Conference Producer at Technology Networks, and I'm pleased to welcome our opening panel today, Under the Radar ADC Innovators to Watch, featuring our esteemed experts Matthew Arnold, Michael Ward, and Hong Wan. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I will hand over to Matthew to open the panel discussion.
Thanks, George, and thanks everyone for joining us today. My name is Matthew Arnold. I'm a recovering journalist and a principal analyst at Clarivate. Clarivate uses data and analytics to help companies in the life sciences future-proof their R&D and speed up innovation to deliver needed drugs, devices, diagnostics, and interventions to patients faster. I'm joined today by Hong Wan, CEO of Antibody Drug Conjugate developer Tallac Therapeutics, and Mike Ward, Global Head of Thought Leadership for Life Sciences and Healthcare at Clarivate. Antibody drug conjugates, or ADCs, are emerging as a powerful platform for delivering highly targeted doses of cancer treatments and potentially other drugs to patients with minimum side effects and toxicity. Large pharmas are very anxious right now to partner with or acquire ADC makers in order to access this technology as they seek to replenish their oncology pipelines with next-generation targeted treatments. ADCs are not new exactly. They're one of the host of therapeutic approaches like RNA treatments or cell and gene therapies that have been in development for decades, but are just now finding their way to patients. Hong Wan has been working in the space for 15 years, I believe, and has seen trial and triumph in it. The first drug based on her company's novel TRAC platform, that stands for Troll-like receptor agonist antibody conjugate, is currently in clinical trials. Hong, tell us a little about yourself and your work, please.
All right, thank you, Matthew, and great to be here with the panel today. Yes, as you mentioned, I have been working in this field for over 15 years. Actually, my tenure in the biotech and the pharma industry actually goes beyond that. I've been working in pharma and the biotech space for about 25 years. Started with as a scientist after my PhD in molecular and cell biology, and then really drawn to the drug discovery and the development aspect, and worked in different therapeutic areas, worked in different stage of development, all the way to phase 2, phase 3 clinical trials, and found my way into the oncology space about 15 years ago, first at Pfizer, and later on at the 2 startup companies, ALX Oncology, where I was the chief scientific officer, and now at Tallac, which I launched about 4 years ago with my co-founders, and being the CEO and the Tallac, and then working with the team and just having a great time, turning the discovery into potentially medicine for patients. So very gratifying.
Thanks so much, Hong. And my colleague Mike Ward has been covering the industry since the dawn of biotech as a journalist and thought leader, and he recently produced a report on ADC companies to watch, which you'll be able to access after this discussion. Mike, tell us a little about yourself and the work you do.
Well, as you sort of say, Matt, I've been sort of covering the industry as a journalist and an analyst for more than 40 years. So I was there at the beginning. I remember when Massachusetts was still basically a parking lot almost. So it's been amazing to sort of see how the industry's grown. At Clarivate, what I'm doing is the thought leadership is actually looking at areas that are developing in the industry that I think are thought-provoking, and the idea is actually to sort of, I guess, kick the tires and sort of understand, okay, how much of it is hype, and actually how much of it is real, and just producing content which is very, very sort of heavily supported by some of the data that we actually have, to just, make people think and, pose questions and just move the conversation on.
Great. Hong, I want to ask you the first question here. I want to start with one that I suspect you know the answers to better than anyone, having been on both sides of the biotech and large pharma divide over your quarter century in the industry. So, what are the challenges that pharma companies face across the development and commercialization life cycle in this space in ADCs? And what should they be considering when they're looking at acquiring and using ADCs?
Yeah, I mean, I think drug development is hard, whether it's for biotech or for larger pharma. Particularly in the oncology space, I mean, there's incredible unmet medical need. The patient is really waiting. However, I think, the biology is complex, the development is complex, and it's actually fast changing. So, think about, we do a clinical trial, starting a phase 3 trial where you have standard care as your control. Where you're doing that trial, the regulators may have approved the new therapy, and that standard care is changed by the time you get your data. So these are challenges, I think, particularly large pharma, when they are dealing with a late stage development and approval, is facing. Obviously, as you mentioned, they need to replenish the early pipeline as well. So I think not only pharma companies in the ADC space, a lot of companies are catching up. And they're looking to acquire approved product or promising product in the early development. So not only they have to consider the acquired asset, they also need to consider their own pipeline in development. And how do you combine the acquired asset within the current portfolio of a pharma company? I think that's definitely a multifaceted question and a consideration for both development as well as a commercialization. Whether that strategy of business development and acquisition can strengthen existing franchise, in particular tumor indication, or opening a new market, or solidify their market position through the new combinations with the acquired asset and their existing pipeline product. I think those are definitely the questions that the pharma leaders, both the development side as well as the business development side, are considering.
Mike, I know you've chronicled the game of catch up that large pharma is playing pretty extensively in this recent report. What did you learn?
Well, yeah, I mean, there are both sort of development challenges that people who want to go down the ADC route have got to face, but also commercialization challenges. So, as Holmes sort of says, that the sort of the complexity associated with the fact ADC development, it's got several components: antibodies, linkers, cytotoxic drugs, and optimizing both these components and the sort of the conjugation process is actually complex and time consuming. Then, of course, there's the challenge of actually identifying the appropriate target antigen, which is overexpressed on cancer cells, but actually has limited expression on healthy cells, because that's absolutely critical for ADC efficacy and safety. There's also the issue, though, is that those ADCs can still potentially bind to healthy cells, which can lead to off-target toxicity, and therefore limit the therapeutic window. And similar to other targeted therapies, cancer cells are actually also can develop resistance to ADCs.
Sure.
So that again would reduce their effectiveness, and therefore people need to be cognizant of it. And then when it comes to commercialization, again, the sheer complexity means that there's a lot of high costs. So therefore there's a lot of benefits as Holmes sort of mentioned about actually sort of having collaborations. The things that particularly sort of small companies need to sort of consider is that the regulatory hurdles. ADCs, they face regulatory scrutiny like others, but actually it's not that clear. For example, when it comes to things like CMC, etc., there's not much guidance at all, if any, and sort of not getting that sort of that regulatory route resolved can actually lead to delay. So that's a sort of there's a commercial challenge there. And then one of the things that you mentioned, the fact it's been around for a long time, but what we've actually sort of seen is in the last 10 years is all of a sudden people are waking up to ADCs again. So we're seeing a lot of excitement. You've got and therefore there are going to be lots and lots of companies competing for market share. And also which is sort of critical is the IP. So it's important that pharma companies evaluate the IP landscape surrounding the ADCs to sort of ensure that they're able to operate freely. So I think, however, with all those challenges, as I said before, as Holmes said, collaborating with other companies or research institutions actually is helping share expertise, resources, risks, and accelerating ADC development and therefore the commercialization.
Mm-hmm. Hong, can you tell us a little more about what you're seeing in terms of the clinical activity in the space and what kind of deal making is happening?
Yeah, it's a really interesting. I guess I'll go back a little bit. My first awareness of ADC was actually when I was at the YS Research. This is before 2010, and personally, I wasn't working in the oncology space, but it was very interesting to know within the company there were development of this new class, antibody drug conjugate. In fact, they actually have the first ADC that ever was approved, a Myotard, which is for the heme malignancy. It's the CD33 clitmicin conjugate that was approved in 2000. And we can come back to this point as some of the challenges and some of the new innovation, because that molecule was approved in 2000. But then Pfizer, after taking over YS in 2009, voluntarily withdrew it from the market because of the toxicity. And then later on, it was resubmitted after Pfizer did additional work based on some of the academics groups' insight in terms of dosing, dose levels, dosing regimen. So Pfizer then went back and resubmitted and got approval in 2017. For new indication, new dosing regimen, and the new doses. So that was a very interesting casing study. That was the first one. And I think now you can see there's, I believe, about 11 ADCs approved. Mike, Mike may have a more accurate number as he done in-depth analysis of the space. But what I'm seeing is from that first one to today, you can see that many of the target for the ADCs are the targets that we were talking about 10, 15 years ago. Mm-hmm. What has really changed is the payload space. Clitmicin, the first one in Myotard, is really toxic, very potent, very toxic. And then you have the next wave of ADC payload that's microtubule targeted, MMAE and auristatin. And that was in the second ADC that was approved, the Genentech's TDM1. And then you have this new wave topoisomerase inhibitor, and then that's in HER2, that's in Trodelvy. And, that's incredibly, I think, interesting, and both from the toxicity as well as activity really broaden the therapeutic index. So I think right now in the current active clinical space, you see activity development of all these payload class. The topo, the MMA, the kind of the microtubule-based ones, and then there's also the DNA agent, PBDs in development, but not all the ADCs are created equal. I think that's one of the challenges for both the developers as well as for the deal makers, because you can see their incredible success, but the Myritas story, I think, also played out in terms of you really can't predict from the preclinical model what's going to work in the clinic. A lot of the activity has to be investigated and then learned through the clinical trial, which to a certain extent is still a bit of a trial and error. So I think the focus for the clinical development of the ADCs nowadays is really to look at how do we broaden that therapeutic index? How do we make a drug that's potent, but at the same time safe and tolerated by patients? So the tolerability is a big issue for the ADC class. I think due to the limitation of and the kind of the imperfect nature of animal models, the true differentiation and the translation has to come from the clinical data. And then the bar in the space is getting higher. We're seeing that assets are being terminated from the pipeline, not because they don't work or they are too toxic, actually because they're not good enough compared to the molecules that's ahead of them. So I think the same time, we're still learning what makes the best design, how to improve and how to broaden the therapeutic index. So I think I'm seeing a lot of activity, but I'm also seeing pharmas are getting more and more selective because there's a lot of substrate to choose from, and then they're really looking for clinical data. A preclinical ADC program, I think, is I think almost a commodity now. You can see a huge number of Chinese companies can just make these ADC molecules very quickly, right? But getting into the clinic and getting that patient data on safety and efficacy, absolutely, that's required now.
Mike, nobody has their eye on deal making in the sector like you. What are you seeing on that front?
Well, as Holmes says, there's a huge amount of interest and activity. Just to sort of come back, across the world, there are 15 ADCs that are approved, and in Cortellis, clinical trials intelligence, we've identified 1,200 ongoing clinical trials. So there's a huge amount of activity. But I think that there's one of the things that has been startling is in the last 5 years or so, we've seen a very, very large upward trend in ADC deals, whether it's licensing, collaborations, or even acquisitions. And some of these are huge tickets. Obviously, the biggest was Pfizer's $43 billion acquisition of Seagen. So Home mentioned about how Pfizer sort of backed off. Well, it's really gone in big here. But we've seen other companies. Gilead has also spent big to get an ADC. And if you look across all the big pharma companies, a lot of them are looking, sort of seeing, right, how do we get access? And of course, what's interesting is that the sort of the key to it is that combining the sort of the monoclonal, the targeting monoclonal antibody with the cytotoxic and the linker technology, and that's complex, and a lot of this actually is housed still within the sort of the the sort of the biotech. So that's one of the reasons why we decided to look at ADC companies to watch, because there's still a lot of scope for innovation, and we know that pharmaceutical companies are looking for that innovation, partly to be able to also provide differentiation, because there's no point in being a me too. So we're seeing it with the acquisitions, but actually sort of also we're sort of seeing big pharma companies getting together and actually deciding, hey, let's do something. So one of the biggest licensing deals that we've seen in the last 18 months or so was the one between MSD and Daiichi Sankyo, where they signed a deal which is worth potentially $22 billion to co-develop, co-commercialize just three of Daiichi Sankyo's ADC therapies against solid tumors. Okay, so we've seen a lot of activity, and I don't see that slowing up in the near term.
I want to move on to regulatory approvals. Hong, we talked earlier about how there's this is really a nascent area for regulation because it's so innovative. You've already, correct me if I'm wrong, you've already won one FDA IND for a drug on your platform. What is the regulatory environment like around ADCs these days? And can you tell me a little bit more about how it differs across key markets? Do you have a sense of that?
I can share some, really like our direct experiences in the early stage, kind of getting to the initial approval to be testing a patient. I think in that sense, I think the regulators, we mostly deal with the FDA. And I think they have a good framework with ADCs. They see a whole number of ADC molecules. They really well aware of the kind of the payload class toxicity to look out to. We have a program that went into the clinic about 2 years ago. And then when we were approaching the FDA in terms of the IND clearance, I think what we appreciate is that they treating our antibody drug conjugate. It's not a cytotoxic payload. It's actually immunomodulatory, very innovative next-gen kind of oligonucleotide payload to activate the immune system. They actually seen this through the traditional cytotoxic ADC lens. So what they asked to do is evaluate, and is in the preclinical setting, the antibody, the payload, and then the conjugate. And I think it's very much approach towards different ADC in the same framework. So I think early regulatory space, they do have a well-established framework. And they know certain, if you come in with a microtubule inhibitor, you will be asked to monitor ophthalmology toxicity and the bone marrow toxicity. If you come in with a topo, you're going to be asked to monitor ILD. So I think regulators are very familiar with the current, as Mike said, there's 1,200 clinical trials. So I think that is, my sense. I think a little bit towards the later stage, I think given the balance of toxicity of activity, I think particularly with FDA, the Project Optimus will have a big impact on the ADC development. I think FDA will be very keen to understand and then encouraging or sometimes require the sponsors to really figure out the appropriate dose levels and the dosing regimen for these molecules. So that may add additional, the companies may have to do additional patient numbers, different dose levels, randomized, compare the different cohorts. I do see that as a trend that will get more intensified for early development.
Okay, Mike, in your reporting, what have you found makes this space challenging for regulators?
Well, it's just, you know, the fact that you've got a large molecule and a small molecule actually in the same place. So that makes it, you know, complex from, you know, both a sort of the product development, but actually also that the sort of the regulatory expectations perspective. You know, as I mentioned, there are no specific guidelines, right, for the CMC part of ADC products. And you know, that is, you know, something that is a challenge for the industry. You know, particularly is that there are just a small number of, you know, facilities at the moment that can actually even handle the sort of the cytotoxic materials. I mean, as Holmes said, some of them are very, very toxic. What we have seen, though, is that there are guidances. So I think, you know, one of the things that really sort of resonated with what Holmes was saying was that the regulators, particularly with these advanced therapeutics, are taking a much more pragmatic view of these medicines, because, you know, it's clear that they are going to have a big impact on patients, and they don't want to be too burdensome. Obviously, they have to make sure that they're safe and they're efficacious, but they are being pragmatic in terms of the way that they're looking at it. But, you know, some guidance just recently, last March, around, you know, clinical pharmacology considerations, again looking at sort of like the small molecule payload, and it's, you know, looking at bioanalytical methods, dosing strategies, etc. So, you know, what we're seeing is that, you know, ADCs, they're regulated as biologics. So, you know, in the US, sponsors submit the listing application through a biologics license application, a BLA, and it's the CDER, the Center for Drug Evaluation and Research at the FDA, that actually oversees the overview of these applications, and they get input from small molecule and large molecule experts. So, yeah, it's a growing or an emerging or evolving art, right, in terms of actually how the regulators are dealing with this. And, you know, there are obviously, you know, ICH and local authorities guidance and expectations that, you know, sponsors would be expected to follow. So I'm expecting that we're going to see more, you know, sort of regulations you know, coming through around ADCs as we understand. But I think that they're there to enable rather than hinder the activities of you know, companies like Holmes.
It's always a delicate balancing act between ensuring patient safety and keeping life-saving innovations flowing to patients, right? Hong, what are the challenges that pharmas face in developing the linker technology? We were talking about that a little earlier, and I know we spoke offline about the Goldilocks problem in terms of toxicity, right? Can you tell us a little about that?
Yeah, so the linker technology is really critical for the ADC. If you think about how the molecules is supposed to work, you want a stable linker, right? That's stable enough in the bloodstream when the drug is first given, that the toxic payload doesn't fall off too easily and then kill all the blood cells. So this refers to off-target toxicity. But then once the ADC reaches the target in the tumor microenvironment in a cancer cell, enters the cancer cell, you want the payload to get released from the antibody so that the payload can then kill that tumor cell it's in. So really, it's a balance of both the stability and the release mechanism. I think, going back to the Mylotarg and then the early ADCs, the linker technology for that, very first generation, they were not super stable. So there's a lot of off-target toxicity that people are seeing in the early molecules. So in the linker space, a lot of focus has been in how do we design linkers to improve that stability. So I think that is improved, is showing some of the later molecules. And then I think another focus, more recent focus, the current focus has been how can we mitigating some of the problem with the payload usually are hydrophobic small molecules to help with conjugating more of the payload on the antibody and the issues with the PK, because if you hydrophobic molecules, your PK is not going to be very good. If you want to have more activity, you want more payloads, but then your PK is going to get shorter. So there's a lot of also technology development trying to solve the problem of improving PK, improving the biophysical properties of the ADC. I think one of the challenges that we still have is I think the toolbox for the linkers are not huge. We still have, generally the cleavable linker and then the non-cleavable linker. And many of the innovation, I think, is more of an incremental improvement of the clinically validated linkers. The true novel linkers, I think I haven't seen a lot of that for the cytotoxic ADCs. And matching the biology of the target, matching the intended MOA. Do you want to release? Is that targeting stromal? Is that targeting, do you want to internalize, or do you want to release, have more of a bystander effect outside the cell? Building that additional specificity that could come from the linker. I think these are all very active areas of research, both for biotech as well as within the, the pharma that has that small molecule expertise working together with the biologics research group to address those issues. So, again, I think one of the challenge is some of these activity, you can't really look at it preclinically, and you have to get into the clinic to really see how this work out. So that, that is a challenge and making it risky for the novel payload, right? Because the risk is higher. So I think a lot of activity goes to optimizing the current linker, but truly a novel linker technology is yet to be seen.
And that explains pharma's increasing preference for investing at the clinical stage in this space. Mike, I know linker technology is one of your favorite topics. What have you learned in talking to these companies?
So I mean, to be perfectly honest, I don't think I could actually add much more to Hom's comprehensive answer. I mean, she's right. Striking the right balance when it comes to stability is absolutely critical, but is equally difficult. One thing that I didn't catch if Hom mentioned it, which I think is also important, is about biocompatibility. So it's important that the linkers don't cause adverse immune reactions or toxicity in the patient. So finding linkers with the necessary biocompatibility is a significant challenge, but also an opportunity. So overall, so linker optimization, complex process, requires extensive testing and evaluation, so that we ensure that we've got that best balance of stability, the biocompatibility, the payload compatibility, and also those crucial cleavage properties. That it lands where it's meant to land.
Right. Hom, what what do you see the next generation of ADCs looking like, and what's the potential to address unmet therapeutic needs?
That's my favorite topic. I love it. I think, I mean, it's really fantastic to see the current ADC become medicine for patients. I remember the ASCO a few years ago, when HER2 data was presented, there was a standing ovation in the entire auditorium. I mean, that's
that, yeah.
fantastic to see that. But I mean, I think still there's a unmet need for the cancer patient, right? And I think going beyond the cytotoxic payload, it's going to have open up a lot of a potential other opportunity, then offer new approaches to address those issues. The next gen ADC class include immunomodulatory ADCs, such as the ones that we're working on at Tallac. I think, I'm also seeing activity, again, this is early, antibody conjugated degraders, antibody conjugated radioligand, to get that really the precision for the patient population. So I think those are super exciting. I think beyond oncology, it's also very fascinating. I think you can think about ways where you can conjugate, particular anti-inflammatory molecules, specifically addressing particular immune cell population for the inflammatory diseases. You can think about, in the antibody oligonucleotide space, we have some early work, but companies like Avidity and Diane are pioneering this for targeting rare disease such as muscle dystrophy. I think even the traditional ADC cytotoxic approach, you can think about pivoting that in autoimmune disease, right? Why not kill the pathological immune cells if you can precisely finding the ones, and then you can deplete the pathological immune cells with the ADC approach, and then that could have an impact in the autoimmune space as well. So I think it's a fascinating, the next generation ADCs, it's going to be several generations and the really interesting applications. Yeah.
Sure. It's hard to believe that we're just at the outset of this, but very exciting.
Yeah.
Mike, anything to add there?
Yeah, well, I mean, just to reiterate that, what Holmes says about, at the moment, all the focus has been on oncology. So, all the ADCs that have been approved are, in oncology indications. We are seeing, however, that there are in the clinic, we're seeing trials for other areas, and I think that is something that we should be looking out for. And then just sort of looking also this sort of some of the deals, and again, one of the reasons why we decided to publish antibody drug companies to watch was actually looking, sort of seeing, right, okay, so what is out there? So it's things like bispecific antibody drug conjugates, right? That's where you actually have two different antigens, sort of simultaneously, right? potentially increasing the specificity and the effectiveness of the drug. Conditionally active ADCs, which also known as pro-body drug conjugates, and they're designed to release their payload under specific conditions, only under those specific conditions. So again, it's a way of reducing that potential off-target toxicity that I mentioned earlier on than the immune stimulating ADCs. Obviously, engineered to stimulate the immune system to deliver the cytotoxic payload, again potentially enhancing the anti-cancer effects. Protein degrader ADCs, targeted protein degradation technology to eliminate target proteins within cancer cells. Again, it offers a potentially new approach to cancer therapy. And then another advance that we're seeing is so-called dual drug ADCs, where they carry two different cytotoxic payloads. potentially allowing for a more potent and selective attack on, again, at the moment it's cancer cells as the target. So we're seeing a bunch of advances in ADC technology, in companies like Hans, that can potentially revolutionize initially cancer treatments, but actually other areas, as other indications as well. And they can, they're also addressing some of the current I guess limitations of the first generation ADCs.
Great. I want to make sure we have time to take a couple questions. The first one, I think, is for you, Hong. What do pharmas need to, or Tallac Therapeutics is working on antibody drug conjugates. Tell us how Tallac's platform is different from other ADCs.
Yeah, absolutely. So I think I touched upon this. So at Tallac, we are developing an antibody conjugate with immune modulatory payload for oncology, and it actually stemmed from our technology platform. So, when we started this back in 4 or 5 years ago, we recognized the traditional cytotoxic small molecule ADCs are going to be a commodity. And we actually figured out a way and develop and optimize technology to conjugate oligonucleotide, much larger modalities to antibody. It's not trivial. You can't just slap a kind of oligonucleotide onto the antibody. So we have a number of technology proprietary improvement and innovation to do that. And then, but we were also looking at the oncology space. Many of our team had a prior experience in the immuno-oncology space, and we're seeing great success of PD-1, PD-L1 inhibitors, but we also have seen a number of failures from other targets on the T cells. We believe that activating the innate immunity, combining with adaptive immunity, is going to be the way to broaden the anti-tumor immune response. So we're choosing Toll-like receptor 9 agonist, which is our proprietary oligonucleotide agonist, conjugating them to different antibodies to precisely deliver them to the immune cells we matter, or to the tumor microenvironment that very specifically. So that's how we think ourselves as really a next-gen ADC with a very differentiated payload. And then the technology can address different modalities and the different therapeutic areas.
Great. And another one for you. There are a number of signaling pathways involved in innate immune response. Tallac is focusing on TLR9. What's unique about TLR9?
Yeah, I mean, this goes back to, I have worked in this space for a long time, and it goes back to some of the comment I made earlier in terms of the difficulty and the limitation to translate from preclinical setting to the clinical setting. There are a number of innate pathways, for example, STING, TR7/8, and others that people have looked at preclinically show great promise, but didn't work in the clinic. So with the TLR9, I think there's a unique scientific biological rationale, but there's also a good literature of a body of literature suggesting it works in cancer patients. This is all from intratumoral delivery of a TLR9 agonist directly to, for example, melanoma lesions and the same response. So what's really unique about TLR9 is there is clinical validation. But the limitation is if you have to inject this into the tumor lesion that's injectable, you're limiting it to melanoma. So our technology, basically we deliver a best-in-class TLR9 agonist in a systemic delivery. So we can actually reach much broader patient population beyond melanoma. So that's we're excited about our current program.
You another one you mentioned, TAC1, the TAC1 compound that you have in trials is an immune targeting TRAC activating B cells. Can you speak more on this approach?
Yes, I think we have a number of programs with this platform. Our lead program, TAC-001, target B cells, activating B cells in solid tumor rather than depleting B cells. You know, if you think of B cell targeting ADCs, you think about those depleting approach in B cell malignancy. So ours is actually opposite. We're activating the memory B cells, the tumor-specific B cells to drive that anti-immune response. Or anti-tumor immune response. And so it's quite unique, but B cells play, I think, a pivotal role in the anti-tumor immune response. B cells are great antigen presenting cells. B cells can produce anti-tumor antibodies. B cell works with T cell in concert to drive the tertiary lymphoid structure in the tumor to really prime the anti-tumor response. So, you know, we're seeing early clinical data demonstrating single agent response in patients who have failed multiple lines of prior IO therapy. So, you know, the clinical data actually really, I think, extend our understanding of the B cell biology in the solid tumor.
Mike, I think this one's for you. What do pharmas need to do to adequately do due diligence when they're weighing an acquisition in this space?
Wow. You spend a long time. I think that the first thing is to actually look at the IP, because yeah, having freedom to operate. You know, we've seen, you know, sort of, companies hoovering up, sort of, platforms, etc. And I think it's also, just looking to sort of say, around that sort of that linker, okay, are you going to get that sort of that perfect balance, right, in terms of the sort of the stability that Holm was looking for. So I think that when companies are looking to identify interesting technologies, I think those are pretty crucial. It's actually sort of having a biocompatible stability is going to think. The other thing, though, is that what in part of the due diligence process, there's one, just looking at the individuals who are involved in those ADC companies in the first place. What experience do they have of actually taking an idea from the bench into the clinic, and maybe they've been successful enough to actually get it into patients as a treatment, but actually sort of looking and sort of saying, right, do they have that ability, and also do they actually have the resources to be able to develop their technology going onwards. Otherwise, I think what's important, it's that what companies are going to have to do, and we've seen it with some of the pharma companies, is that they actually invest quite heavily in helping those biotechs actually get the resources they need to develop it. But it's I don't think, apart from the looking at the sort of how cool is the linker technology, how good is other targets, etc., that they're going for, I think most of the due diligence is actually not dissimilar for whether they were going for ophthalmology products or gene therapy products or whatever. It's much more of the same.
Maybe I can just interject one point. I like, Mike, what you're saying, and I think earlier you made a point of this is a very complex biologics, right? And it's big molecule, small molecule, protein, and then cytotoxic payload all in one. The CMC is critical. I think pharma looking at this, they will pay a lot of attention to, can you make this drug? What is the process? Can you scale it up? They're thinking about commercialization. Right? What's the cost of goods? All those questions. That's also very, kind of an essential part of the due diligence process.
Hong, one more question for you. You have a tumor targeting as opposed to immune cell targeting approach within the Nectin 4-track TAC-003. how is this different from TAC-001 and also from Nectin-4 ADCs?
Yeah, great question. So I think our approach, our platform is pretty versatile, right? We have the payload that activate innate immunity through the TR9 pathway, but by conjugating it to different antibody that drive either targeting the immune cell, such as the TAC-001, or a tumor antigen, such as a Nectin-4 in the TAC-003 case, we can address different needs in different tumor indications. So we actually initially started looking at the tumor antigen target. We look at the HER2. We published some data, preclinical data with HER2, but we very quickly realized, it's a very crowded space. You have to think about also the biology. A lot of HER2 positive tumors are not really immunogenic. So we turned our attention to Nectin-4. We liked it very much. We selected several years ago, working on this, and it has a really high expression in bladder cancer. And the bladder cancer, if you think about it, the first approved immunotherapy BCG, right? So this is a sensitive tumor type to immune modulation. And so now we have a clinical candidate, TAC-003. Preclinically, we published the data and the CT and the AACR before. It's very differentiated from ADC. It's not toxic. Nectin-4 ADC have issue with on-target toxicity. It's expressed in skin. It's expressed in GI tract. So you do get those on-target skin tox with PADCEV, which is approved in bladder cancer. So we don't see that toxicity. We see very differentiated safety profile in non-human primates. And because it's not the same payload, it has a very different resistance mechanism. We don't believe you will have the same resistance mechanism as Nectin-4 ADCs. And it can also work in lower expressing Nectin-4 expressing settings because it doesn't need to internalize into the tumor cell to kill the tumor cell. So we see very good activity preclinical setting in low and medium Nectin-4 expressing tumor, much better than the ADC, the traditional cytotoxic ADC. So this opens, I think the possibility of working in other indications where Nectin-4 expression is low. It's very heterogeneous. Right now, the cytotoxic ADCs hasn't really worked, such as the non-small cell and the breast cancer. So we're excited about this program as well.
That is exciting. Unfortunately, that's all we have time for today. I want to thank Hong and Mike for the fascinating discussion, and I want to thank all our viewers for joining us today. The Clarivate report, Companies to Watch, Antibody Drug Conjugates, that we mentioned earlier, is available for download on clarivate.com, and we will be sharing a link with registrants as well. And we look forward to watching the progress of Tallac's innovative ADCs and hopefully seeing them in patients soon. Thanks so much again to everybody, and over to you, George.
Massive thank you once again to Matthew, Mike, and Hong. But unfortunately, as you mentioned, that's all that we have time for today. That was a fantastic panel discussion and a great way to open day 2 of this event. And we are so incredibly grateful to all of you for showcasing your expertise with us today. It's certainly an exciting time in the ADC field, and we cannot wait to see the latest developments. Just as a reminder to all in our audience, any questions that we didn't have time to get to will be sent to the relevant speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. And that just leaves me to say thank you once again to our experts, and we will be back soon with the next talk.